Hello, everybody. My name is David Hayes. This research project was performed under the mentorship of Dr. Laura Robertson. The aim of this research project was to isolate, identify, and characterize growth on substrates and antibacterial activity of fungi local to Jefferson County, West Virginia. With the exception of JCF50, all of our filamentous fungi were isolated from the surface of fruiting bodies of wood decay fungi. JCF50 was captured in the microbiology lab at Shepherd University. JCF 9, 10, and 12 were isolated by Hunter Palmer and species identified by Paige Vanneman. JCF 50 was both isolated and species identified by Allison Beverlin. Once pure cultures were isolated for each of our filamentous fungi, we identified the species of each isolate by sequencing the ITS region of the ribosomal RNA genome. JCF 9, 10, and 12 were all identified as Trichoderma atraviridae. JCF 20 and 22 were both identified as Mucornidicola. JCF 21 was identified as Geotrichum klebani. JCF 50 was identified as Aspergillus niger. With the exception of JCF 50, we were initially trying to isolate the wood decay fungi, but ended up isolating the parasites off of the fruiting body. Growth of the 3 Trichoderma atraviridae isolates was evaluated previously by Hunter. I characterized the growth of my 2 Mucornidicola isolates and Geotrichum klebani isolate on 4 different substrates: cherry sawdust agar, malt extract yeast peptone agar, potato dextrose yeast agar, and dog food agar. Shown here on the x-axis, we have intervals of time in days, and on the y-axis, we have the diameter of the fungal colony in millimeters. 90 millimeters is the maximum growth of the fungal colony, as that was the diameter of the Petri dish. JCF 20 was able to reach 90 millimeters on MYPA, PDYA, and DFA by around day 4, and was unable to grow significantly on the cherry sawdust agar. JCF 22 was able to reach full plate on MYPA, PDYA, and DFA between days 2 and 3, and was also unable to grow significantly on the cherry sawdust agar. JCF 21 grew best on the MYPA, but also grew well on the DFA and the PDYA. JCF 21 was completely unable to grow on the cherry sawdust agar. The growth noted is from the initial inoculum. This experiment was performed concurrently with the species identification. We were initially trying to isolate the wood decay fungi they were isolated off of, but instead isolated the fungus from the surface of the fruiting bodies. Because we initially thought we had isolated wood decay fungi, we expected them to be able to grow on the cherry sawdust agar. Since we actually isolated fungi off of the surface of the wood decay fungi, it makes sense they were unable to grow on the cherry sawdust agar. We assessed the antibacterial capabilities of JCF 9, 10, 12, 21, and 50 in competition with a panel of bacteria, which included E. coli K12, Staphylococcus epidermidis, and Bacillus cereus. Cultures of the bacteria were allowed to incubate overnight in nutrient broth and were then diluted tenfold. We swabbed a lawn of the bacteria on potato dextrose agar. An approximate 6 millimeters square of fungal mycelium from the outer edge of the fungal colony was inoculated onto the center of the plates. They were then incubated at 26 degrees Celsius, and the diameter of the zone of inhibition was measured at 20 hours. Each assay was performed in triplicates. Pictured here on the left, the yellow square is the fungal inoculant. The yellow circle is the area of fungal growth. The white circle is the area of no bacterial growth, and the blue circle is the area of bacterial growth. Our reported zone of inhibition is equal to the diameter of the area of no bacterial growth minus the diameter of the fungal colony. Shown here on the right is an example of a zone of inhibition of Trichoderma atraviridae in competition with Bacillus cereus. Shown here are our no fungus controls of Bacillus cereus on the left, Staphylococcus epidermidis in the middle, and E. coli K12 on the right. The opaque growth of Bacillus cereus is easy to see in these photographs. However, all 3 of the bacterial species grew as a confluent lawn across the surface of the culture medium. Shown here are pictured examples of the assay. On the left, we have the no bacterial control. Then we have the in competition with Bacillus cereus, in competition with Staphylococcus epidermidis, and then on the right, we have in competition with E. coli K12. Panel E shows that JCF9 was able to exhibit a zone of inhibition while in competition with Bacillus cereus, but not while in competition with Staphylococcus epidermidis or E. coli K12. Shown here is JCF12 JCF10, I apologize, with the no bacterial control on panel A. Panel B shows in competition with Bacillus cereus. C shows in competition with Staphylococcus epidermidis, and D shows in competition with E. coli K12. JCF10 was also able to exhibit a zone of inhibition while in competition with Bacillus cereus, but not Staphylococcus epidermidis or E. coli K12. Shown here is JCF12 with the no bacterial control while in competition with Bacillus cereus, Staphylococcus epidermidis, and E. coli K12. JCF12 was also able to exhibit a zone of inhibition while in competition with Bacillus cereus, but not Staphylococcus epidermidis or E. coli K12. All three of our Trichoderma atraviridae isolates were able to exhibit zones of inhibition in competition with Bacillus cereus, but not the other two bacteria tested. JCF21 and JCF50 were unable to exhibit antibacterial activity against the bacterial panel. Shown here on figure A and E, we have the no bacterial controls. B and F, we have in competition with Bacillus cereus. C and G, we have in competition with Staphylococcus epidermidis. And then D and H, we have in competition with E. coli K12. In conclusion, we showed that Trichoderma atraviridae is able to inhibit the growth of Bacillus cereus observed across different strains of Trichoderma atraviridae. Other strains of Trichoderma atraviridae are being investigated for possible biocontrol of fungal pathogens. We have shown that our Trichoderma atraviridae strains isolated from the surface of wood decay fungi also have potential to be used for antibacterial control. Thank you.